Dear students, today we are going to discuss plastids. Plastids are membrane bound organelles which are found in plants and algae. The term plastid was given by Haeckel, and these plastids are responsible for manufacturing of food, which is done by chloroplasts in plants. They are even responsible for storing food, maybe carbohydrates or proteins or lipids. And these plastids, they have pigments which may change at certain stage of the growth, and the color of the cells may also change because of this change of pigments. As we see that when the vegetables like tomato, which is not ripe, then it's green, but later when it becomes ripe the color is red. So that is because of the change of pigments in the cell. Plastids are semi-autonomous organelles. That means they have DNA the same way as mitochondria has, and of course this DNA is smaller than the nuclear DNA, has very less genes than the nuclear DNA, but of course this DNA codes for many important enzymes of the plastids. Plastids arise from the precursors called as proplastids. These proplastids are very small organelles which are membrane lined. They do not have pigments, so they are colorless. And the lamellae inside, lamellae is the internal membrane network that is scattered, that is not organized, as we'll discuss in chloroplast. They are scattered lamellae, which is internal membrane system which lies scattered in the matrix of the plastids. These plastids may also contain starch and also the DNA, as I discussed, which is naked. Now there are different kind of plastids which are present in plants and algae. These can be differentiated on the basis of presence or absence of the pigments, such as chromoplasts, which are colored, gerontoplasts, the leucoplasts, the uncolored, and the chloroplast. Let us discuss the chromoplasts first. Chromoplasts, they are the pigment containing plastids which have dominantly the carotenoid pigments present in them. They develop from the leucoplast, which are without photosynthetic pigments, or the chloroplasts also may sometimes change to chromoplast, because the chlorophyll which is present in chloroplast, that will degrade and carotenoids will become the dominant pigments in these organelles, which eventually we call them as chromoplast. So chromoplasts are found in flowering plants, in aging leaves and fruits, that which I discussed. Then when the fruits they become ripe, then their color changes from green to some red or yellow. That is because the chloroplast that will break down, and other pigments like carotenoids, they will be synthesized more. So carotenoids present in chromoplast, it gives yellow, orange or red colors in leaves and the flowers or the fruits. Chlorophylls are not present in chromoplast. These chromoplasts, because they provide color to the structures which contain them, therefore they help in attracting pollinators, or they even help in attracting animals to eat fruits for dispersal of seeds also. So they have a role in helping the plant in attracting the pollinators for pollination, and the animals for dispersal of the seeds. Gerontoplasts are yet another kind of plastids which are actually the chloroplast at the aging process. These chloroplasts, when they are aging they are aging at that time, they start converting to some other structures, they start transforming into some other structures, and they no longer play the role, or they have the role in photosynthesis. That means they do not have the pigment also, and they transform into some other kind of structures. So The leucoplasts, which are non-pigmented plastids, that means they have no color. These leucoplasts, thus, because they lack pigments, they will be present in such structures of the plant, such parts of the plants which are non-photosynthetic, like the roots. These are majorly responsible for storage of starch, lipid or protein. They are also the sites where amino acids and fatty acids are converted. And these leucoplasts, they possess thylakoid lamellae, but they do not have grana. That means the lamellae, that is the internal structure, internal membranes in the stroma of the plastid, and when they are stacked one above the other, then they are called as grana. So lamellae are present in leucoplasts, but grana is absent. Those leucoplasts which store starch are termed as amyloplasts. Those leucoplasts which store proteins in them, they are called as proteinoplast or aleuroplast. The leucoplast, allyloplast or lipidoplast, they are the ones which store the fats and oils which are needed by the plant. Now we'll discuss the next plastid, which is the chloroplast. The chloroplast is that organelle, that plastid which contain photosynthetic pigments, the chlorophyll and carotenoids. Because of these pigments, the chloroplasts are capable of capturing the sunlight, and this sunlight is changed into chemical energy which in which oxygen is released, which is a byproduct. Oxygen is released from water. Therefore, they are the organelles which have role in photosynthesis. Chloroplasts are found in all green plants and algae. They were first described by Leeuwenhoek and Grew, and the name has been given by Schimper. The number of chloroplasts in the organisms that may vary in the chloroplasts in the cells of an organism may vary from one organism to another, but usually in algae it is fixed. The number of chloroplasts in a cell is fixed for a particular species, but from one species to another the number may change, but it stays fixed for one particular species. Their shapes may also change. In higher plants, because these plastids are in the shape of these chloroplasts as biconvex or plano-convex shape, but in algae we find variable shapes, such as it is plate-like in Eulothrix, it is cup-shaped in Chlamydomonas, it's ribbon-shaped in Spirogyra, stellate-shaped in Zygonema, and reticulate shape in Oedogonium. These shapes have been already studied by you in the first semester when you studied biodiversity while studying the alga. Now, these chloroplasts, they are found in the mesophyll cells of the leaves of the plants, or some other modified, sometimes stems may also modify to be photosynthetic. So those cells will also contain chloroplast, but in leaves it is present in the mesophyll cells. The size of chloroplast in higher plants, the average size is 4 to 6 micrometer in diameter and 2 to 4 micrometer in thickness. Now, we'll have a look at the detailed structure of chloroplast. Like mitochondria, chloroplasts too have two membranes, the outer membrane and the inner membrane, and both these membranes, they enclose a space called as intermembrane space. And while we see the structure of chloroplast, this is the outer membrane, this is the inner membrane, and in between here this is the intermembrane space like mitochondria. The outer membrane in chloroplast is the similar more permeable membrane than the inner membrane as in mitochondria. The outer membrane, it has many channels which allow bigger ions or molecules to pass through. The protein porine, these porins are channels which allow at least 5,000 Dalton size particles to pass through them. Though everything is not allowed to pass, but still this outer membrane is more permeable than the inner membrane. The inner membrane is lesser permeable and it is more specialized, and only certain selected material can pass through this particular. In addition to regulating the kind of material that will pass in and out of the chloroplast, they have major role. This internal membrane has major role in regulation activity. The fatty acids, lipids and carotenoids are synthesized in the inner chloroplast membrane. Then the next is the stroma. When we see the structure again, this in the compartment which is inner to the inner membrane, it has a fluid matrix which is called as stroma. The stroma of chloroplast, it contains the thylakoid membrane system. If we see predominantly this system, these are actually the membranes. This network is found in stroma. The DNA of chloroplast is lying in stroma. The RNA that it codes, the DNA codes, that lies in stroma. Many enzymes and proteins that lies in stroma. Plastoglobuli, the plastoglobuli that also lies in stroma. Pyrenoids, they lie in stroma, and the eye spot or the stigma that lies in stroma. You can read some more details about stroma. Here the DNA in chloroplast, that is again naked and circular like the prokaryotic DNA, the arrangement in prokaryotic cells, and it may be present in many copies in the cell. Ribosomes are 70S, that means they are primitive kinds of ribosomes like the prokaryotic ribosomes. The enzymes which are present in the matrix, they are majorly the enzymes responsible for synthesis of photosynthetic pigments, photolysis of water, photophosphorylation, carbon dioxide fixation, the synthesis and degradation of starch, and synthesis of lipids. So these all processes, metabolic processes, they take place in the stroma of the chloroplast. Therefore the enzymes for these all activities, they are present in the stroma. Now the lipid droplets called as plastoglobuli, these are also present in stroma. In certain algae, pyrenoids are also seen. Pyrenoids, these are refractile regions, specialized regions of the matrix which store the starch. These are present in some algae. In some motile algae, for the perception of light, some photosensitive organs called stigma or eye spots are also present in the stroma, and these are majorly these eye spots, they contain photoreceptor pigments, flavin or rhodopsin. So this is about the stroma. The next lies in stroma is the thylakoid system. This thylakoid system, this thylakoid system is a membrane system. These membranes, they will stack somewhere, then they go free, then they again stack somewhere, then they will continue in the stroma. The thylakoid membranes which are stacked, these stacks are called as grana, and singular is granum, and many stacks are grana. And the thylakoid membrane which is running through the thylakoid, through the stroma, they are called as the stromal lamellae or stroma thylakoids. So if we see, this is a structure of a grana. Grana, here we see that it is, these are the stacks of the thylakoid membrane. The region inside is called as the lumen. Here it is filled of some kind of matrix, and this part of the thylakoid membrane which is facing each other, जो दबा हुआ part है, which is not exposed, एक थाइलाकॉइड का दूसरे थाइलाकॉइड के सामने जो सैंडविच्ड पार्ट है, सामने का जो पार्ट प्रेजेंट है, दिस पार्ट इज कॉल्ड एज द एप्रेस्ड पार्ट, जो प्रेस हुआ है एक दूसरे के साथ. ये जो साइड में इसके पार्ट है, ये साइड में जो इसकी जगह है, दिस वन, दीज आर कॉल्ड एज द मार्जिन्स. हियर यू सी, ये मार्जिन्स है, ये मार्जिन लिखा है. इफ इट इज नॉट क्लियर टू यू, दिस इज मार्जिन. और ये जो सबसे बाहर वाले पार्ट है, जहां बाहर अब स्ट्रोमा लाई करता है, ये वाला पार्ट भी जो है, दिस इज कॉल्ड नॉन एप्रेस्ड पार्ट. ये वाला पार्ट और ये मार्जिन्स, ये दोनों नॉन एप्रेस्ड पार्ट बनाते हैं, जो कि प्रेस्ड नहीं है. इस पूरे को ग्रेना स्टैक बोलेंगे. ये जो ग्रेना स्टैक से स्ट्रोमा में जो थाइलाकॉइड लैमेली है, उसे स्ट्रोमल लैमेली कहेंगे. सो this arrangement is important because some of the parts, some of the machinery of photosynthesis for photophosphorylation, they are present in the non-appressed parts. Some are present in the appressed part and some are present in the margin. This is here you see grana, then stromal lamellae, फिर ये जाके फिर से stack होगी, which is called grana. Same way so many grana they are formed. I was talking about the appressed and non-appressed portions of chloroplast. These thylakoid membranes contains 4 major complexes, photosystem 1 and photosystem 2, that we know, which are involved in photophosphorylation, along with cytochrome B6 and F complexes and coupling factors. Coupling factors are the ones which are ATPases, जो कि ATP बनाते हैं. ये सारे के सारे जो हैं हमारे photophosphorylation के components हैं. These are present in thylakoid membrane because we know that the seat of photophosphorylation or the light phase of photosynthesis are the thylakoid membranes of the chloroplast. These photosystem 1 they occur in stromal thylakoid and the non-appressed part. Stromal thylakoid मतलब ये है stromal thylakoid जो कि स्ट्रोमा में जा रहे हैं, और non-appressed part कौन सा है, ये margins और ये this one. यहाँ पे photosystem 1 जो है वो मिलता है, and photosystem 2 is present here in the appressed portion, जो कि धंसी हुई, जो एक दूसरे के सामने फेस करती हुई जगह है, यहाँ पे appressed portion में है the photosystem 2. Cytochrome B6 and F complexes, they occur both in appressed and non-appressed portions, so they have a distribution throughout the thylakoid membrane, but the ATPases they are present in non-appressed portions of the grana. And the photosynthetic pigments are also associated with the photosystems, that we know, because photosynthetic pigments are the only ones which capture the sunlight and they are the part of photosystems. So this is how the structure of structural components of chloroplast are arranged within the organelle. Now we'll discuss the functions of chloroplast. Chloroplasts, as we know, they are the organelles which are the ones responsible for photosynthesis. They form sugars, and oxygen is also released during photosynthesis, which is made available to us, and this light energy that is changed to important chemical energy, which serves as source of energy for other living organisms. These organelles also store starch. जो भी यहाँ पे carbohydrate synthesize होता है, temporarily in plant cells it's temporarily stored in the chloroplast, but in algae these are permanently stored. Because of the light reaction, NADH2 and oxygen, which is due to the photolysis of water, that all is the role of chloroplast, because that happens with at the thylakoid membrane, production of NADH2, evolution of oxygen, and due to the photophosphorylation along with NADH2, ATP is also synthesized. So chloroplast is responsible for the synthesis of ATP by photophosphorylation process. The conversion of PGA into different sugars, phosphoglyceraldehyde into different sugars and starch, that is because of that happens at the chloroplast. So chloroplast are the cooking site for these are the kitchens of the green plants. Then synthesis of lipids and storage of fats, that's also happens in the chloroplast, and fats are stored as plastoglobuli, which are present in stroma of the organelle. Chloroplast also provide photosensitivity to some algae because they have stigma or eye spots present in them, which help the algae to sense the light from where the light is coming. So that is about the functions of the chloroplast. Also, in some organelles, in some cells, chloroplast, they change into chromoplast, where these chromoplast will become dominantly rich in carotenoids, which will help in making the plant parts colorful, therefore attracting the insects or animals, which help in the dispersal of seeds or pollination from the flowers. So these are the functions of chloroplast. What are the marker enzymes for chloroplast? That means कि वो कौन से enzymes हैं जिनसे हम कहीं पे पता लगा सकते कि chloroplast organelle यहाँ पे present है कि नहीं है. वो marker enzymes होंगे chloroplast के लिए. So that is the rubisco. We know rubisco is the enzyme which fixes CO2 for photosynthesis. And NADH, glyceraldehyde 3-phosphate dehydrogenase, these two enzymes, they are the ones which are the marker enzymes for chloroplast. Now, how chloroplast that is made? Plastids may arise de novo from pre-existing plastids. Any plastid may arise de novo. De novo, that means by themselves, or they may arise from an existing plastid. So chloroplasts may origin from proplastids, or they may come into the existing chloroplast may divide to give rise to more than one chloroplast. So this is how biogenesis of chloroplast that occurs. This is all with this lecture. Hope you all are studying well.